IBD essay. Since 2000, the global prevalence of inflammatory bowel disease, IBD, which includes ulcerative colitis, UC, and Crohn's disease, CD, has risen, now affecting about 1 in 200 people in Western countries. These chronic inflammatory conditions differ in symptoms and immune responses, with mucosal immunology playing a central role in disease development. IBD arises from a complex interplay of genetic, environmental, microbial, and immune factors, leading to persistent inflammation and tissue damage. Current research focuses on understanding these mechanisms to improve treatment strategies now and in the future. Difference between UC and CD. Inflammatory bowel disease, IBD, includes two main types, Crohn's disease, CD, and ulcerative colitis, UC, which differ in inflammation patterns, location, and complications. CD causes transmural inflammation with skip lesions anywhere along the GI tract, often affecting the small intestine and leading to fistulas and nutritional issues. UC involves continuous inflammation limited to the colon and rectum, primarily affecting the mucosal layer and increasing colorectal cancer risk. Both share symptoms like diarrhea and cramping, but UC is more associated with bloody diarrhea and anemia. Environmental factors such as smoking impact the diseases differently, and genetics also play a significant role, especially in CD. Treatments for both include anti-inflammatory and immunosuppressive drugs like anti-TNF agents, with CD often requiring more aggressive early intervention and UC focusing on mucosal healing to prevent cancer. Pathogenesis of inflammatory bowel disease, IBD. Inflammatory bowel disease arises from a complex interaction between the immune system, genetic predisposition, microbial dysbiosis, and environmental influences. Mucosal surfaces, including the gastrointestinal, respiratory, and urogenital tracts, act as vital barriers to the external environment, equipped with specialized immune defenses to maintain tolerance while preventing infection. These include immune cells like dendritic cells, macrophages, and T lymphocytes, along with secretory IgA and the resident gut microbiota. Secretory IgA, produced in mesenteric lymph nodes and Peyer's patches, neutralizes pathogens at the mucosal surface, while a balanced microbiome is essential for immune regulation. When this equilibrium is disrupted, especially in genetically susceptible individuals, it may contribute to the development of IBD. Genetic factors significantly contribute to IBD risk, especially in Crohn's disease, where monozygotic twins have a 50% concordance rate compared to 16% in ulcerative colitis. Key genetic variants include mutations in the NOD2 gene common in Western CD patients, which impair bacterial clearance and trigger inflammation, and polymorphisms in the Illinois 23R gene, affecting Illinois 23 signaling important for Th17-driven mucosal inflammation in both CD and UC. The immune system, particularly innate and adaptive responses, is central to IBD pathogenesis. Innate immune cells such as dendritic cells, macrophages, and intestinal epithelial cells exhibit abnormal activity in IBD, contributing to persistent inflammation. Adaptive immunity is also dysregulated, with T helper cell subsets including Th1, Th2, and Th17 playing distinct roles. In CD, a Th1-dominated response drives the production of interferon gamma, IFN, TNF, and interleukins such as Illinois 12 and Illinois 17, stimulating further cytokine release from APCs. Conversely, UC tends to exhibit an atypical Th2 profile characterized by Illinois 4, Illinois 5, Illinois 19, and particularly Illinois 13, which disrupts epithelial barrier integrity through effects on apoptosis, tight junctions, and epithelial restitution. A deficiency in regulatory T cells, which normally suppress excessive immune responses, further aggravates the inflammation. Microbial imbalances also play a major role in IBD. Individuals with IBD often exhibit a reduced abundance of protective bacterial species, such as firmicutes and bacteroids, along with increased levels of pro-inflammatory microbes. This dysbiosis is associated with compromised epithelial barrier function and increased bacterial penetration of the mucosa, intensifying the immune response. In UC, a thinner mucus layer is frequently observed, potentially facilitating greater microbial invasion. In CD, immune cell infiltration causes not only inflammation but also structural changes such as thickened intestinal walls, fibrotic strictures, and mucosal edema due to the healing of chronic lesions. Disruption of the intestinal barrier is another critical factor in IBD. Tight junction proteins that regulate paracellular permeability become dysfunctional, allowing microbial products to cross the epithelial layer and activate underlying immune cells. This heightened permeability enhances immune cell infiltration and amplifies mucosal inflammation. Moreover, plasma cell activity is altered, with increased production of IgG and its subclasses, e.g., IgG1, IgG3, and IgG2 in CD, replacing the usual IgA response. Since IgA works with lysozymes to inhibit microbial colonization and antigen penetration, this antibody class switch impairs mucosal defense and promotes further inflammation. Cytokines and chemokines released by activated immune cells also recruit neutrophils, which degranulate and release reactive oxygen species and leukotrienes, causing epithelial damage. During the acute phase of IBD, especially in the lamina propria of affected segments, large numbers of macrophages and T cells accumulate, intensifying the inflammatory response. Environmental influences further modulate IBD pathogenesis. Dietary patterns high in refined sugars have been shown to disrupt gut microbial composition and promote inflammation. Smoking is a well-known risk factor for CD, while antibiotic use has been linked to dysbiosis by decreasing microbial diversity and altering gut homeostasis. In conclusion, the development and progression of IBD result from a multifactorial interplay involving genetic predisposition, immune dysregulation, microbial imbalance, epithelial barrier dysfunction, and environmental exposures. These factors converge to create a chronic, self-perpetuating cycle of inflammation and tissue damage in the gut. Immunology and therapeutics. The traditional view that Crohn's disease is Th1-driven and ulcerative colitis is Th2-driven is an oversimplification, as both conditions share overlapping inflammatory pathways targeted by advanced therapies, involving diverse immune subsets like Th17, Th9, Th22, innate cells, and B cells, with insights from monogenic IBD highlighting common final inflammatory mechanisms in polygenic disease. Current treatments. Five ACEs, which are anti-inflammatory medications, have been approved for the treatment of UC and have been shown to induce remission, however they have a minimal impact in CD. They work by suppressing the production of pro-inflammatory cytokines, decreasing neutrophil chemotaxis and decreasing mast cell activation. They are generally well tolerated, with anti-inflammatory effects shown in UC patients. However, they are ineffective for CD and pose possibility of mild side effects as well as rare nephrotoxicity. Corticosteroids are also anti-inflammatory medications which have been shown to induce remission for CD and UC patients, however they are unable to sustain long-term disease control. These work by affecting the gene transcription by interacting with DNA to activate or repress the transcription of target genes involved in inflammatory processes. Also, they affect the transcription of pro-inflammatory mediators such as cytokines Illinois-1 and Illinois-6. By suppressing these pathways, corticosteroids reduce inflammation. However, their long-term use is associated with risks such as osteoporosis and metabolic complications, which limit their use in IBD management. Targeting the immune system is a core strategy, focusing on established inflammation. Anticytokine therapy in IBD. Anti-TNF therapies play a crucial role in treating IBD by targeting TNF, a central cytokine involved in the inflammatory cascade driving chronic intestinal inflammation and tissue injury. Drugs like infliximab, a chimeric antibody, and adalimumab, a fully humanized antibody, are widely used anti-TNF agents that exert their effect partly by inducing apoptosis in cells expressing membrane-bound TNF, which appears essential for their therapeutic success. However, the development of antidrug antibodies, immunogenicity, can reduce their effectiveness, and this is often managed by combining anti-TNF therapy with immunosuppressants such as azathioprine. Additionally, therapeutic drug monitoring helps maintain adequate drug levels, particularly important in patients with active disease where intestinal loss of the medication may occur. Unlike these antibodies, etanercept, a soluble receptor TNF inhibitor, has proven ineffective in IBD, highlighting that neutralizing only soluble TNF is insufficient for disease control. Blocking TNF may also lead the immune system to compensate via alternative inflammatory pathways, such as an increased Illinois 23 response, which could contribute to disease relapse. Biologic therapies for IBD include not only anti-TNF agents but also inhibitors targeting other pro-inflammatory cytokines like Illinois 12/23, as well as integrin antagonists that modulate immune cell trafficking. TNF itself is a potent driver of inflammation, activating immune cells, promoting tissue damage, and facilitating pathological processes like neoangiogenesis within the gut. The mechanism of anti-TNF drugs involves reducing inflammation by blocking TNF activity on various gut cells, including macrophages, T cells, and fibroblasts, thereby diminishing immune activation and encouraging apoptosis of pathogenic T cells. This comprehensive immune modulation underlies the clinical efficacy of infliximab and adalimumab. The failure of etanercept in Crohn's disease further emphasizes the importance of targeting membrane-bound TNF rather than soluble forms alone. Overall, anti-TNF therapies remain a cornerstone in managing IBD by effectively suppressing inflammatory responses and preventing tissue injury, often serving as a first-line advanced treatment option depending on clinical factors and cost considerations. Illinois 12 and Illinois 23 blockers. Blockade of both Illinois 12 and Illinois 23, via the P40 subunit, is clinically effective. Blocking Illinois 23 alone, via the P19 subunit, appears more effective in CD, suggesting preserving Illinois 12 signaling may be beneficial. Illinois 23 is vital for the maintenance and terminal differentiation of Th17 cells. Anti-trafficking therapies. Vedolizumab and anti-integrin therapy in IBD. Integrins.